Thanks everyone for joining today. I'm excited to have the opportunity to be back with you again. Thank you, Parker, for inviting me. And I want to talk to you today about kind of another field I'm really interested in, and it's called Nutrigenomics. And basically what that is, we're going to get more into it, is the interaction between nutrients, what you get from your diet, your supplements, your nutrition, and your genes. And how do those interact to create health, disease, and what can we do about it? So let's jump right in. I'm going to start by doing a brief overview of genetics, epigenetics, SNPs, which are single nucleotide polymorphisms. And most of you, I think, have already heard a lot about this, so I'm not going to go into too much detail. I'm just going to reintroduce it so we have it fresh in our memory and so we can understand how it relates to our metabolome, the biochemical milieu of our body, and how that relates to health creation, which is what we're all about. And I want you to understand the potential of nutrigenomics and precision medicine to guide individual lifestyle recommendations for you. So how can you take this data and make it actionable? And we're also going to talk about how can you get this data so you can make sense of this for yourself. Quick disclaimer I need to put in all talks. I am not your doctor, even though I am a doctor, so this is not professional medical advice. Always be sure to work with your own health practitioners. So Human Genome 101, we have over 3 billion base pairs, and these are pairs of nucleosides, which are proteins, and A for adenine is paired with thymine, they always go together. C, cytosine, is paired with guanine, so AT, C, and G. And they're wrapped, as you know, in a double helix and coiled together, a single double-stranded DNA coiled together into chromatin, which is the protein of really finely coiled DNA, and that's coiled again to make chromosomes. And chromosomes are the main packaging material for DNA in the nucleus of our cells. We have 23 pairs, one set from each parent. 22 are kind of regular chromosomes, and one pair is sex chromosomes. So us guys have one X and one Y, and females will have 2 X chromosomes. There's a side, I'm not going to get into the detail, but there's something called mitochondrial DNA, which is inherited just from your mother. So dads have nothing to do with that DNA. And a metaphor I'm going to use throughout the talk is thinking of this kind of like a recipe book. So in the recipe book, your DNA is the actual book, the recipes, the content that is in the book. But as we know, we can buy books, read them, and don't necessarily use it. So at the end of the day, what matters is which recipes are made and how they're made. So we're going to get into that. SNPs, I'm sure many of you have heard of SNPs, single nucleotide polymorphisms. So these are point mutations in one of those letters, A, T, C, or G, and they usually lead, the ones we're going to talk about, lead to a difference in functioning of a key enzyme in the body. Some of them are related to changing the functioning of a protein. Some are related to how we react to different medicines based on liver enzymes. So there's a whole plethora of SNPs, and this research is really at its infancy. We're just learning, we're gathering big data in unprecedented levels at a very cheap rate, and trying to interpret how do we connect that data to health outcomes. So there'll be a lot more to learn in this field coming up. Back to SNPs. When we're thinking of types of SNPs, some of the lingo that's used is wild type. That's the natural form of that, the natural sequence of DNA that most people have. Then you have heterozygotes. So that's a plus/minus. It means you have one of your chromosomes will have that mutation and the other one doesn't. And then homozygous, plus/plus, where you have 2 copies of the single nucleotide polymorphism. So a common one you'll hear about is MTHFR A1298C. What does that mean? So MTHFR is an acronym for the enzyme, methylene tetrahydrofolate reductase. 1298 is the location on the DNA. And then A in the beginning is switched to a C. So A is the wild type on the left, C is the mutation. So if you have that mutation, it would be heterozygote would be A/C, homozygote would be C/C. And then, just because we have a gene, it doesn't mean that those genes are actually going to be used. It's like having a recipe book that you just never open the page to that recipe. And so this is where the field of epigenetics comes in. And it's a really exciting field, as I'm sure you've seen in other talks, because even if I have genetic risk factors for a certain disease, doesn't mean I'm going to get that disease. Our genes are not our destiny. We can activate or silence genes based on the information we give our body. That comes from nutrition, food, supplements, toxic load, things like heavy metals and plastics, environmental and work influences, or stressors. Because as we know from last session, stress changes the biochemical milieu of our body moment to moment. So the key question is, how do I know, what does my genome tell me about my unique risks and opportunities? How can I use that info to optimize my genetic expression? Just like our recipe book. So this is a diagram kind of reviewing what we've talked about so far. You have your double-stranded DNA, and it's coiled together in chromatin, and that makes up chromosomes. And through the process of epigenetics, there's something called a histone group, a protein, that will wrap around certain genes, making them essentially unreadable, inaccessible by your body. And that process of methylating genes is the mechanism of epigenetics. We're going to talk a little bit more about methylation and the methylation cycle in the future. So kind of tag that in your memory for now. And so it's more, obviously our health is more than just a gene. So let's look at some of these other words you might have heard thrown around and exactly what they mean. At the end of the day, our health is about much more than just our genome, or even which genes are being expressed. Our genes are turned into RNA, and those are carried outside the nucleus to something called a ribosome, which, think of it like a 3D printer inside each cell. And the ribosome will make proteins based on that recipe. Those proteins can be turned into lipoproteins like LDL, apolipoprotein A or B, amino acids, nucleotides, neurotransmitters, hormones. Everything in our body is made through this process. And so our genome is our DNA. Which genes are transcribed, so which pages in the recipe are read, is called our transcriptome. From that, which proteins are actually made is called our proteome. And looking at the whole milieu moment to moment of all of these different proteins is our metabolome. And the metabolome is what leads to functioning of our body, either creation of health and well-being or creation of disease. So when we're thinking about nutrigenomics, breaking that down, nutrition and genome, we're looking at what we take in. So food is essentially information. Our body is designed to break it down into component molecules and absorb it. There can be a problem with that if we don't have the right gut bacteria to break it down. So it's more than just our genes. There's also probably 10 non-human cells for every single human cell in our body. And they obviously interact. So the food is taken in based on SNPs that we have, our genetic differences. Certain genes will be expressed or not expressed. We're going to talk about a principle called Le Chatelier's principle of how we can alter the functioning of enzymes based on our dietary input. And nutrition can also affect epigenetics. So that's where you see methylation and creation of those histones, where the DNA is wrapped around a histone and silenced. And this has an impact on our health. This slide specifically about cardiovascular risk. So personalized medicine, we're learning that there is no one-size-fits-all approach. We can't use the same algorithms to apply to every human being because we're all so different. And we're just at the infancy of looking at how different we are. In medicine, there's a group called ICD-10 that tries to make a list of every diagnosis. And I think the whole system is flawed of trying to take an individual and package them into a group. It's good for coding and billing, but it's not necessarily great for health because there's so many different types of diabetes, for example. So our goals are to help you understand your own uniqueness. And then, as I said in the beginning, turn that into actionable data to change your lifestyle to create better health. So let's look at some examples. First, I want to talk to you about how you can learn more about your SNPs, especially the relevant ones. Probably the easiest is starting with 23andMe or Ancestry.com. It's the same gene chip, so you don't need to buy whatever package is cheapest. I think there's like a Ancestry package or a health and disease package. That just means 23andMe is only releasing certain info to you. Either package will let you download your raw data, basically a bunch of letters of ACTG, into a zip file. And you can go to a site called Promethease. I listed there for about $12, less than 10 minutes. It'll upload your data and connect it with SNPpedia, which is essentially an online database of all the different SNPs. And I put a screenshot there. In the very top column, there's a drop-down menu, and you can change that to different sort of panels that are in Promethease. Probably the best one for what we're talking about today is the NutriHacker SNPs. And you can see there's 63 SNPs in that database. And so Promethease will give you a report of your unique SNPs from that database, so you can use that to change your lifestyle. You can also find some of this data with traditional labs, True Health Diagnostics, Genova. They have a product called Genovations, Quest, LabCorp. And the third secondary analysis tool is called Genetic Genie, and they can use 23andMe data. And they'll give you a profile that's pretty user-friendly on methylation and detox. And those are two of the more actionable areas for nutrigenomics. So I just put a screenshot. This is my Genetic Genie profile of methylation from my 23andMe. And so you can see the gene, the variation, the IDs, the location on the gene, the alleles, the different letter differences are there, and then the result. Again, plus minus is heterozygous, plus plus is homozygous, and then the wild type is green, is minus minus. So I told you we're going to talk about methylation, and it is a complicated pathway, but very important. All of the things in blue squares are enzymes. And all the enzymes with a star by them have relevant SNPs that we know about that alter the function of those enzymes. And to make it a little easier, these blue arrows will point to key things you might have heard of. Some things that can be checked in blood levels. As you know, with mental health, serotonin and dopamine are critical neurotransmitters. And let me see if you can see my mouse here. So for example, let's say you have a slow variant because of a mutation of MTHFR. That will process this cycle in this direction from BH2 this way to BH4. So If this is slower and not processing as fast, you're going to be moving BH2 to BH4 more slowly, right? And you'll have less of this available, and you'll have more of this available. It'll kind of be backlogged. And so that can have a positive impact, with more dopamine levels, more serotonin levels. They can have a negative impact too with production of homocysteine, which is an Alzheimer's risk factor and cardiac risk factor. And so one of the ways we typically will treat a cardiac MTHFR mutation is by adding in a form of folate called methyl tetrahydrofolate. And when it already has a methyl group attached, it can help to push this enzyme and optimize its efficiency in the biochemical process that it does. You can also see cofactors for some of these enzymes. ZN is zinc, B12, zinc, B6, SAM, S-adenylyl methionine, magnesium, B2. You'll want to make sure if you have some of these slower variant mutations that you're getting plenty of the cofactors the enzymes need to do their job well. Another really key important variation in SNPs that we usually test for is called apolipoprotein E. You might have heard about this in Alzheimer's research. It's probably where it gets the most press, but it's related to a lot of things, and we're going to get into that. It's on chromosome 19, and it's kind of a cholesterol taxi in your body and especially in your brain. Important for lipid metabolism, for your digestive process, specifically around fats, and inflammation in both your body and brain. It is broken down based on your genetic variant into a protein called ApoE3, that's the normal one, the wild type. ApoE4, that's about 15% of the population. And then ApoE2, about 8% of the population. E3, again, is the wild type. No special diet recommendations here. If you happen to be E3 and have high blood pressure, there's data that quercetin, that's a plant flavonoid, you can get in a supplement, or it's in, I think, black tea, red wine, different berries, you can get it through food, will help to decrease blood pressure, specifically with this ApoE isoform. If your inflammatory markers are high, quercetin will also decrease TNF alpha, which is a key regulator of inflammation. ApoE2, on the other hand, this is the least common, their bodies tend to process sugars and simple carbs in a way that worsens their negative impact. We know they're bad for us, but they're especially bad for people with ApoE2. So what to do? Limit them as much as possible. And ApoE2 people have decreased efficiency at absorbing fat from their diets. So even if they eat fat, it doesn't make it into their body. So they need extra fat in their diet. And we always prefer healthy fats, olive oil, canola oil, avocados, sardines, one of your best fish. There's a new, new to me, the sardines at Costco and olive oil. Super simple. One little tin has 1,700 milligrams omega-3, very sustainable for the environment. So I think we should be more like the Portuguese, eat more sardines, especially if you're ApoE2. Nuts and seeds. And you may need higher doses of the fat-soluble vitamins. So vitamin D, E, A, and K. And omega-3 levels. Because we usually get omega-3 from diet. And we can't absorb that fatty acid as well. We have to take increased doses. The good news with ApoE2 is it's actually protective against Alzheimer's disease. So all in all, I think it's a good isoform to have if you're an E2. E4 is the one that gets the most press. And what it does is it causes an increased efficiency of absorbing fats in our intestine. So there's two fats that are especially bad for us: saturated fat and trans fats. Saturated fats traditionally are found in animals. Think of dairy, butter, cheese. If you look at a meat before you cook it, all the white is fat. So you can see bacon is full of all that solid white material, ribeye steak, fatty hamburger meat, chicken skin, all that's saturated. On the plant side, coconut oil, palm oil, macadamia nuts are some of the more common vegan saturated fats. Trans fats, hydrogenated, anything with the word hydrogenated or partially hydrogenated in the nutrition facts, like if you look at Skippy peanut butter, for example, usually found in processed foods, cookies, pastries, fried foods, Cool Whip, fake butters, that sort of thing. So you want to have zero intake of those, especially if you're E4. And then because they're better at absorbing fat, they're better at absorbing the fat-soluble vitamins, so they often will need less high doses than someone with ApoE2, for example. Now, E4 people also will have an overactive pro-inflammatory response and an underactive anti-inflammatory response. That can lead to a lot of problems because most chronic disease is caused by overactive inflammation, chronic inflammation. Alcohol and tobacco really worsen this with E4 people. So if you're E4, you're doing somewhere between 4 to 5 times the level of damage to your body and brain if you use alcohol and tobacco. So useful to know, especially with even people who don't have drinking problems, who drink socially, may decide to stop drinking or drink a lot less if they're ApoE4. It's important to have a plant-based, colorful diet. The colors in plants are phytochemicals that are antioxidant, so will protect against inflammation. ApoE4 people are specifically responsive to cardio. And so as they do more physical activity, specifically cardio, their overall disease risk and inflammatory markers get a lot better. So when you're trying to look for high leverage strategies, you can see how much your genotype matters and the SNPs you have matter. Because they're better at absorbing fat, they need a little bit less omega-3 and ALA supplements. That comes from a mice-based study. And in the brain, they have reduced clearance of beta amyloid. Beta amyloid is a toxic byproduct produced in our brain from just normal everyday life. And it needs to be cleaned out at nighttime. If it doesn't, it starts to develop disease, mainly Alzheimer's disease. And we can see if people die by accident, the disease process of Alzheimer's starting about 40 years before you start to see symptoms of Alzheimer's. So with the age, it usually starts over age 65. We're talking your lifestyle after age 25 is really relevant for your Alzheimer's risk. And I wanted to start with these two mutations. There's many more. There's a lot related to detox. There are SNPs related to how your body processes different pharmaceutical medicines, and all sorts of fun facts like how you process caffeine. So much more to learn here. And as I said in the beginning, research is really at its infancy. So stay tuned. There's going to be a lot more. And in terms of actionable steps now, start by knowing your data. Use some of the resources I mentioned early, and they'll give you a lot more that explains each and every SNP. And then after you understand that, connect with a professional, whether it's your physician, a nutritionist, a naturopath, who can help guide you to make a plan of what do I do? These are my genes. This is my current lifestyle. This is where I want to be. What do I need to do differently? And then do it. So that's all. Thank you so much for your time and attention. Look forward to talking with each of you individually at the mentor office hours. Take care.